So last time when we were talking about our class, our lecture, our main focus was on basically understanding the nervous system and the main components, the main cells that are involved, etc. Now our focus for this for this lecture is going to be on central nervous system, which we basically learned going to be brain and spinal cord. So most of the lecture today for the central nervous system is going to be solidly on the brain, since brain is the structure that's going to do most of the actual activity. And a spinal cord, as we can see later on, it's more like a highway between the brain and the rest of the body. It is extremely important because, again, if that highway is cut, basically you lose the communication with your brain. It's also responsible for the reflexes in our body. So basically, for instance, if you touch something hot and then you pull your hand away from that surface, that reflex actually comes from the spinal cord rather than your brain. The pain you may feel as a result of that comes from your brain, but the actual reflex of pulling a limb away from your from a danger, closing your eyes, things like that, these are reflexes that are associated with your spinal cord rather than your brain. So let's go ahead and get started. So when we look at your brain, I'm going to just show you on the picture because it's a little bit easier to demo on a picture rather than on slide. So you have this kind of color coding on this image right here. So we have the largest part of the brain, which is about 80% of your brain, depicted in yellow. This is called the cerebrum. On the back of the cerebrum, on the inferior part of the brain, you have this orange structure called cerebellum. Not to be mistaken, the names are kind of close to each other. And then if I literally open up the brain and look at the center of the brain, you see this purplish structure right here. And this purplish structure is, as a whole, is referred to as the diencephalon. So they kind of showed you the zoom in version in 2 shades of purple. Diencephalon itself is made up of 2 structures you may have probably heard of sometime in your life, maybe the name in the passing. One is the word thalamus, which is this kind of egg-shaped, oval-shaped structure you have here. And then the smaller structure underneath it called hypothalamus, which is this triangular structure right here. Please do note that even though this is not labeled, you've probably seen it in your brain anatomy when we were doing it last week for your lab. This little thing hanging right here, that's called the pituitary gland. And that's extremely important for your endocrine system, and it is under the control of your hypothalamus. The last part of your brain, which is the 4th part of the brain, is what you can see depicted in green section. So the 3 shades of green you have here. And then again, the other structure that you have right here is the kind of, what am I spelling exactly? Sorry. Pituitary gland? Okay, yes, so you got it. So pituitary gland. Yes, thank you, Michelle. So I was like, what did I say that I need to spell? That's not here. Thank you, guys. Okay, so for brain stem, as you can see, it's kind of like the stem of a flower. It's holding on the brain, right? Now, brain stem itself has 3 segmentations, and each of these segmentations do a very particular function for us. And they also, as you can see, they have these yellow structures sticking out of them, and they are called the cranial nerves. So basically the nerves that are going to control the activities of different parts of your face or your body. And we'll talk about them very briefly later on. Now, brain stem is again made up of midbrain, which is the first part, right underneath the diencephalon. This kind of the bulging structure you see right here called the pons. And the darker shade you see right here called the medulla oblongata. Now, logically, if you continue the medulla oblongata farther down, as you can see down here in this picture, that basically becomes your spinal cord. So please do note that brain and spinal cord are not two separate structures, especially when we look at them in terms of embryonic development. It forms as one structure and then advances to form the brain and spinal cord. And again, please guys, if you do have any questions, type them in the chat box for me. Now, brain development is really fascinating. It's one of the structures that actually starts to grow at a very early stage or early ages of the embryonic development. So when we start to draw or grow the nervous system, in this case, central nervous system, what we actually start with a structure that is only the cell bodies of the neuron. If you guys remember the neurons, they had that kind of chubby structure that has the nucleus and all of the organelles. So when a nervous system is developing at the very early stages of the embryonic development, before the 4th week reached, so the first month of pregnancy, if you want to think about it, this neural tube, all it is a collection of cell bodies that doesn't even resemble a neuron. Now, these selections of cell bodies form a structure called a neural tube. And I'll show you this picture. This is all it is. This is all the nervous system is. Just literally a tube made up of cell bodies. So it's not a fascinating nervous system. Like a lot of time I tell my students, central nervous system, neurons, and they kind of think about an actual brain. No, it's not like that. But here's the fascinating thing, guys. By the 4th week of development, so again, a month into the pregnancy, you have what we call primary brain vesicles, which you can see there's a significant difference in terms of both the size and the complexity. So the brain is actually starting to form now. So during primary brain vesicles, you're forming 3 segments, and you can see them as 3 separate size bulges, 1, 2, and 3. These 3 structures are called prosencephalon, or forebrain, mesencephalon, or midbrain, and rhomboencephalon, or hindbrain. So basically, forebrain means the part that is in the front, right? It's the part that we can see. Midbrain, again, in the middle, and hindbrain typically in the back or hiding. So it's not something that is visible to us as much. So after, and I want to go back to this slide previously, after only just 1 week, guys, so from the 4th week to the 5th week of development, you'd go from primary brain vesicle to a secondary brain vesicle. And here's the complexity, guys. You're going from 1 week, 7 days, guys. This amazes me every time I think about it. Primary brain vesicle, secondary brain vesicle. 4th week of development, 5th week of development. So again, you can see that the brain is starting to fold itself. It's starting to become an actual structure that may eventually resemble an adult brain. Now, one thing I want you guys to note here is that you can see that these 3 primary brain vesicles we started with are now becoming a smaller or individualized structures for themselves. So for instance, prosencephalon becomes your telencephalon and diencephalon, etc. For our class, guys, the one that I do want you to focus on is, first of all, knowing that we start with the neural tube, going to the primary vesicle, going to the secondary brain vesicle, and eventually an adult brain, the structure that we will see in all of our brain right now. I also want you guys to know that the primary brain vesicle has those 3 parts, the prosencephalon, mesencephalon, and rhomboencephalon. However, I don't want you guys to know this breakdown right here. So the breakdown I have to the secondary brain vesicle, it is not something that I'm going to test you on. Is it something to know? It is a good idea to know it. It will help you later on in your education, but it's not something that you require to know for this class right now. What you do want to know is the following. These primary brain vesicles that I have listed here, what do they develop into an adult brain? So let's take a look. prosencephalon, which is again majority of what we call forebrain, the anterior aspect of the brain, or I should say the superficial part of the brain, are going to be your cerebrum, which was again what we see, what we visualize. And then the other thing that's hiding underneath the cerebrum is your diencephalon. Remember the purple structure, the oval-shaped structure right here, which was basically your thalamus and your hypothalamus. So these structures are all associated with your prosencephalon. It is not listed here, but I do want to just kind of mention it to you right now, that the retina of your eyes, basically the part that gives you your ability to see, also comes from the prosencephalon, which is basically, if that doesn't develop, the person would be identified as blind. Mesencephalon, which is known as midbrain, kind of self-explanatory, give rise to your midbrain portion of the brainstem, which was the middle section, sorry, the first part of the brainstem. And then anything else that basically is left over is associated with your rhomboencephalon, which is pons, medulla oblongata, your cerebellum, which was that chunky part in the back of your brain. And then again, notice that the continuation of the neural tube becomes your spinal cord. So in an exam, guys, expect questions like this. What primary brain vesicle does cerebrum associate with? So I'm going to just type it in your chat box so you guys get the type of questions. Now it could be associated with any structures, but just to give you again an idea. What primary brain vesicle does cerebrum originate from? And in this case, the answer would be your forebrain, which is also the same name as prosencephalon. And again, please, guys, know both names, because I can give you just the name forebrain, or I can give you the name prosencephalon. Questions or clarification on this, guys? Okay. I have shown you not only in the picture, but I also have it broken down here for you. If you guys do want it to have it as a written statement, but they are here, so you can review that as well. Okay, so let's continue. Now, when you look at your brain, guys, and again, it's not something that you can visualize in the models that we have seen. If you look at the brain model, or if we were actually in the class and we looked at the sheep's brain, to see the color differences that are depicted here, called gray matter and white matter. However, if you're looking at a fresh brain from either a cadaver or sheep's or a cow, whatever specimen we're looking at, you would be able to see a very minor shade of or color difference between the two. And I want to just kind of show you on a picture. So right here, notice that the edges of the brain, so anytime you have basically that cut that goes deep into the brain, or the superficial aspect of the brain has a darker color to it, and we refer to that as the gray matter. And then what you see underneath that, that's as a lighter shade, those are called the white matter. So gray matter and white matter, basically the coloring. What you also see is within your white matter, so this is your white matter, you have a spots of what we call gray matter again. So you have gray matter, which is superficial, followed by underneath it, the white matter, and then patches of gray matter inside the white matter. So what is the gray matter? So gray matter is basically what we identify as the neuron cell bodies. They can also be short, non-myelinated axons. So tell me, guys, what can we find inside the cell body of a neuron? What does the cell body contain? You guys remember from our last lecture? Anybody? Participate. No? It's called cell body or soma. Okay, you have your cytoplasm, correct? You have no axon and dendrites come off of your cell body. But what is your cell body itself contains? So the chubby part of a neuron, it contains what? Organelles, correct. Organelles are nucleus. And what is your cell body responsible for making? So now that it has all of the organelles and nucleus, what is the most important thing it makes? How does a neuron communicate, guys? It does create energy. It does create ACH, but what is the general terminology we use for ACH? ACH is a category of what? It does contain mitochondria, which is an organelle, correct? Neurotransmitters, thank you. So it actually, when you think about the cell body, which is the most important part of a neuron, you contain within that cell body your nucleus, which is basically your control center, and all of your organelles. And why do I need all of those components? Because I want to be able to produce neurotransmitters like ACH, and that is going to be used for communication from one neuron to neuron. So basically, if a neuron cannot make neurotransmitter, it is useless. There's no point to having it. So the cell bodies are concentrated mostly on the superficial aspect of the brain, which is again called gray matter. And then myelination. What does the word myelination imply, guys? You guys remember that? What is when a neuron is myelinated? What does that imply? What do I have for that neuron? Starts with an I. The answer starts with an I. One word. It is insulated. Thank you, Solma. So it is insulated. Your neuron is insulated, which basically means that the electrical impulses that are going to pass through the axon portion of the neuron is going to transfer very rapidly. So if it's considered gray matter, those are non-myelinated. Basically means they have very little insulation. That doesn't happen as often. Most of what we call gray matter are cell bodies, but it is good to know that it could also represent the non-myelinated neurons as well. So what is all this space underneath that? What is that, all of the white matter that's underneath it? And that would be the myelinated axons. So mostly myelinated axons would be identified as what we call a white matter. So when you guys are looking at this, again, this is typical exam question. What is gray matter made up of? What is the white matter made up of? So you should be able to distinguish what that is called. Now, quick note, guys, when we talked about gray matter that is found in the cerebrum, which is, again, this part of the brain, the visible part of the brain that we have. The gray matter, I'm going to type this in the chat boxes so you guys have that. The gray matter of the cerebrum is referred to as cortex or cerebral cortex. And I think I have it in the future slides, but just to make sure that everybody's on the same page. That superficial aspect, again, you can see it as a darker color, is specifically for the cerebrum. It's called cerebral cortex. Oh, I just noticed that it says it's referred to as cortex, and it has it here too. Any question, anything needs clarification so far, guys? Are you with me? Are you studying for your lab exam? I hope that's not the case. Because I'm pouring my soul out here. Okay, so let's continue. So when we look at your central nervous system is extremely important for your functionality, right? Because basically it controls what you would be doing in your environment, controls your muscles, controls your heart, your breathing. Everything in your body is controlled by your central nervous system. So the question is, what do I do to make sure that my nervous system is protected? So we already kind of know the idea. We have basically bony structures around both the brain and the spinal cord called the skull and the vertebral column. So those are the common structures that are going to provide the most protection. In addition to that, however, and we talked about it, I briefly mentioned it, I think a few lectures ago, we also have layers of protection that surrounds the brain itself, brain and spinal cord itself, called meninges. And I think a lot of you guys may not have heard of the word meninges, but you have heard of the medical condition called meningitis, because we did have an outbreak maybe like 3 or 4 years ago, before this whole COVID happened and everything went down the drain. But we had an outbreak I think in San Diego was one of them, and Berkeley was the other one, where they basically had number of students being infected by meningitis in the dorm rooms. So meningitis is basically inflammation of the protective membranes that are surrounding your central nervous system called meninges. Now let's look at these meninges. Again, there are 3 layers of meninges or protection, and they are referred to as dural matter from outside moving into toward the brain, arachnoid matter, so middle layer, and pia matter, which is the layer that literally sticks on the surface of the brain and gives it a very glossy look. So again, working from outside in, you have dural matter, arachnoid matter, and pia matter, the 3 layers of your meninges. So what do you need to know about each of these layers? So dural matter, which is the closest one to your skull, and I want to just kind of show you the picture here. So dural matter is the closest one to your skull right here. It's a double membrane. So you have this membrane and this one. They're both dural matter, so the white layer. And you have a gap between them, which is what you see in blue. It's called dural sinus. So here is, again, kind of introducing this. This is your skull right here. Here is your brain tissue right here. And these are the 3 layers of meninges. So the double layer of meninges called dural matter with a space between them called dural sinus. So what is the importance of this? So this gap, which is called dural sinus, is extremely important because it contains what? Blood vessels that eventually drain into your jugular vein. So what does that mean? So basically, this is the part where all the blood vessels from the brain that collect deoxygenated blood, collect that blood and eventually return it back towards your heart. So if somebody basically has some sort of trauma that causes the skull to be damaged, that skull can get pushed into the dural matter, cause damage to these blood vessels, and cause internal bleeding into the brain, which is something that we don't want to have. And it's basically called a hematoma, right? A hematoma is bleeding into the, in this case, your brain. So again, dural matter is a double layer. Between that is dural sinus, and dural sinus is where your blood vessels basically collect blood from your brain and return it back to the heart through the jugular vein. The second layer we have, which is the layer you see right here, is the arachnoid matter. So again, notice that the arachnoid matter also has a space that is kind of looks segmented. It's kind of like, to me, kind of looks like a beehive. So this middle layer right here, that's your, what we call a subarachnoid space. So that space right here with all the little openings, that called subarachnoid space. And then the innermost lining, which is literally covering the surface of the brain is the innermost layer right here called the pia mater. So if you're looking at your brain and you're looking at the meninges, sorry, I should say brain and spinal cord, the space which is called subarachnoid space, the space we have right here, is filled with a fluid, and we have talked about that last time, called called cerebrospinal fluid, or CSF. And you're going to see this again in just a minute, but I'm just going to type it for you guys. So if you see CSF, again, it stands for cerebrospinal fluid. Basically a fluid that flows around the brain and the spinal cord. So if I ask you again, where do I have the cerebrospinal fluid? Your answer should be around your, or within the subarachnoid space. Subarachnoid space is the space that we have the CSF flowing. So 3 layers: dura mater, arachnoid mater, pia mater. Each of them will have their own importance. So as I mentioned, meningitis, the medical condition, one of the things that it kind of scares me about meningitis in general is the fact that it can be caused by either a virus infection, a bacterial infection, or a fungal infection. So you really don't have that say, oh, it's a cold, it's a bacterial infection. It could be caused by a variety of reasons. And all it is, meningitis, all it is, anytime, again, learn this right now, it makes your life a lot easier. Anytime you hear itis at the end, itis means inflammation. So meningitis basically means inflammation of your meninges. So if you take a look at the picture we have right here, here is a healthy brain tissue right now. Your meninges are pretty healthy. They're not inflamed. Right here, you have the scenario of meningitis, where basically the 3 layers have swollen up, and that caused that inflammation has affected the brain. Understand one thing, guys, here, that when you are dealing with meningitis, if you have a brain, which is a soft tissue here, and you have your skull here, right? A skull is not gonna move, right? So if there is inflammation in the space between those two, what is that inflammation going to do? It's gonna affect the soft tissue, which is your brain. And that's a really bad thing. That inflammation can also affect the brain tissue directly, which basically means that the person is going to have problems doing a lot of the activities. Yes, the meningitis does cover it. So hopefully, yeah. So when we're talking about, sorry, not meningitis, meninges cover both the spinal cord and your brain. So when you have meningitis, which again can be a result of many, many different bacterial viral infections or even fungal infections, you are at a severe risk of not being able to recover. Because, and I'm going to just add that it's not on your PowerPoint, a lot of the symptoms of meningitis when you start it is very similar to a typical cold. So you have a headache, you feel stuffy, your neck hurts. So it's not a very obvious, I guess, sign that you have meningitis, if you want to put it that way. And by the time you notice that there might be a problem, it would be typically too late. So in this case, I think the chance of you having a viral infection, viral infection that causes meningitis, your chance of survival is higher compared to if you have the bacterial infection for meningitis. And basically what they do is they pump you with antiviral, antifungal, antibacterial medication, and basically hope that that medication helps and the inflammation goes down. Yes, actually, a lot of people die from meningitis. I think that the numbers are 1 in 2 if you have bacterial meningitis, and then 1 out of 3 if you have viral infection. Yeah, I mean, the chances are still good. Again, it depends on when they catch it. So if the doctor is pretty good and it's maybe have diagnosed a lot of cases of meningitis, maybe if there is an outbreak, then they can easily test it and they can start the treatment. But kind of think about that when you hit a point of no return for meningitis, then you're done. It doesn't matter. If the damage to the brain has already happened, then it already happened. But I'm glad that your mom recovered. Yes. But again, in the case of like the meningitis outbreaks that we had in the schools, there are actually students that have passed away as a result of that. So they kind of downplayed it because, again, Berkeley, UCSD, they didn't want that to be out there as much. But I don't know if you guys recall it, it did make it to the news and it was kind of sad. And then they started all those campaign about don't go to the things, don't go to the the dorms, and like if you're sick, don't hang out with people, let that pass, because you don't want to pass it to other people, not knowing you have meningitis, and you spread it to other people because it passes like a common cold or common flu. So here's the thing: how do you diagnose if somebody does have meningitis? You're welcome. So how do you diagnose if somebody has meningitis? So you do that by doing what the process called a spinal tap. So you can put the patient in one or the other position, and basically the lying position or sitting position. If you're a woman and you had an epidural, this is the same position as the epidural. And they're gonna go between your vertebra and they're gonna inject it is contagious, yes. And they're going to inject a needle, I should say, pass a needle between your vertebrae, and they're going to take a sample of your cerebral spinal fluid. So it's supposed to be CSF, not SCF. And then what they're going to look for the infective agent. So they're going to look to see if there's a bacteria or fungus or virus in that space. Second thing they look for is sugar levels, which is surprising. Like, what the heck? Why do I care about sugar level? So think about it. If you have an infection, those viruses, those bacteria, those funguses, what do they need? They need food, right? What's the best source of food that you have in your body? Sugar. So they basically use that sugar, which causes low level of sugar in your cerebral spinal fluid. So really dangerous? I believe so. I'm not as confident in my answer, but it is an infection. So yes, the babies can get it. Actually, now that I think about it, one of my best friends says actually she had it. She was a premature baby, and yeah, she got it when she was under a year. So yeah, you can get it, and you're again at a higher risk if you are a premature baby, like any other condition, unfortunately. Okay. So any other question, guys? And I did let somebody in, so if you haven't signed in, please do so. Okay, so moving on, guys, no questions? Sorry, I'm like, somebody's like yelling in my house. I'm like, what the heck? Okay, so cerebral spinal fluid is again the fluid that surrounds your brain and your spinal cord. The main goal of this, if I ask you guys, why do I need that fluid around my brain, is to provide cushioning and protection for the nervous system. It also transports nutrients and chemicals. So we'll talk about that in just a minute. But the way I describe that protection and cushioning, a lot of people is like, wait, why would having some fluid around my brain going to protect me? So here is the scenario, guys. Imagine your brain is a tennis ball inside a can, which is your skull. Now imagine there is no fluid inside this can and you shake the can, which is basically represent trauma. So the brain, your ball, is gonna bounce off inside your skull and it's gonna hit the inside of your skull, which means not lubrication, which means basically you are damaging the surface or the superficial aspect of your brain, which has all of the cell bodies and important things. And if those blood vessels, if the actual tissue is damaged, the chance of you recovering from that is pretty low. Now, what if we take the same scenario, we fill the can of the can, which is your skull, with water, and then we put the tennis ball back in there. Now what you have is the brain is now, or the tennis ball is gonna be buoyant inside, which means if I shake the can, the ball is not gonna bounce hard inside the can, right? Because the water takes a lot of that force away from it. So if you do suffer from trauma, having that fluid around your brain basically allows the trauma to be less significant on the brain tissue. Now, does that mean you can still get brain damage if you have severe trauma? Yes or no? Can you get brain damage even if you have that fluid around your brain? Yes, like many football players, right? Many people who have suffered from car accidents, right? So just because I have this fluid, it doesn't mean I can hit my head in a wall and say I'll be fine the next day, right? So you are still susceptible to what we call concussions, right? You're still going to be susceptible to things like, what do you call it? Hematomas, right? Bleeding in the brain. But your chance of having severe brain damage from minor cases of trauma is significantly less. Okay. Questions, guys? Comments? Anything needs clarification? Awesome. Okay. So where is this CSF comes from? Where is this fluid comes from? First of all, this fluid that we're talking about is a very small amount of fluid. It's about 150 milliliter, which is a little bit less than like a bottle of water, the small water bottles that you have. So around your brain and things, a lot of people like, they think it's like this giant watery smushy things around your brain. No, it's not. It's a relatively small amount of fluid. And the really cool fact is that it actually gets replaced every 8 hours. So in the 24-hour time that you have, the fluid that flows around your brain gets replaced 3 times, which is amazing to me. So kind of think about how sensitive your brain is, that you really need to change that fluid completely every 3 hours. Sorry, every 8 hours, not 3 hours. So where is that actual fluid come from? It comes from cells or a structure called choroid plexus, which is basically a bunch of capillaries and cells that are found inside cavities of the brain. And they're responsible to basically produce and recycle your cerebrospinal fluid. Now we mentioned choroid plexus is made up of cells and capillaries. So what are these cells called? This is something I mentioned to you guys the last class meeting when we were talking about supporting cells of the central nervous system. And the cell that I mentioned for production of CSF were called ependymal cells. So ependymal cells are basically these cells you see in the grayish color. So here's my capillaries, basically. That is my blood vessels. Here is the ependymal cells. And this thing as a whole is referred to as the choroid plexus. So one of the first characteristics of the ependymal cells you notice is the little tiny hair-like structures that are sticking out of it, which are called cilia. Sorry, didn't come. I hate that. So what you will have is basically, here is your blood. Here's your blood that has all the nutrients, the plasma. It has glucose, fats, sodium, potassium, calcium, chloride, etc. They come in, and some of that fluid, not the red blood cells, not these ones that you see, not the white blood cells, just the fluid portions and anything that's dissolved in that is going to come out of the capillaries. They're going to pass through the ependymal cells, which are the cells that are basically covering the capillaries, and they're going to get into your CSF and form your CSF. So again, CSF is formed from the filtrate containing glucose, oxygen, vitamins, ions like sodium chloride, etc. Anything that your cells have used at, the purple things are your nucleus for the ependymal cells. So anything that is considered to be waste, so something that are unwanted, the waste product like carbon dioxide, urea, uric acid, things that your cells have produced, your neurons have produced, and basically it shouldn't be a part of your CSF, are going to go through the ependymal cell, as you can kind of see, go through the capillary bed, and then get away from your brain. So you have that recycling happening here again. Waste goes in through the ependymal cells, go to your capillaries, and anything your brain and your tissues need come out through the ependymal cells and gets used by your cells. So it is extremely important to remember that the fluid that we call CSF comes from choroid plexus. The main cell types that are associated with them is ependymal cells. And again, this is the structure that is the cell. And you're asking again, as the purple circles, those would be your nucleus. Anytime we have cells and there's something purplish inside of it, remember that's going to be the nucleus. That's the easiest way I remember it. So let's go ahead and continue. Now, we have mentioned that the cerebrospinal fluid is flowing inside our meninges, right? More specifically within a space called the subarachnoid space, right? Between the second and third layer of the meninges. But do we have that fluid other places within our brain and spinal cord? And the answer is yes. Within your brain, guys, you have cavities that are referred to as the ventricles. And again, this word, if you notice it, is literally the same word that we used for your heart, which basically your bottom chambers of the heart. Now, we have cavities inside the brain. We have a total of 4 of those called ventricles. And then another place we can find CSF in our nervous system is, if you think about your spinal cord, that kind of like this snake-like structure that's going down the back of your body. Imagine there's a hole right inside of that, right? That hole is called the central canal. And the central canal also contains your CSF. I just signed in somebody. If you guys knew, please make sure you sign in your name so I know that when you join the class, please. So if I ask you again, locations for CSF, be specific. The locations would be the subarachnoid space, your ventricles, there are 4 of them in terms of your brain, and then you have the central canal that's associated with your spinal cord. Okay, so check, make sure we're good with time. So what are these ventricles and central canal looks like? We're just going to look at them on a picture, and then you guys can go back and study the actual slide on your own time. So in this picture, first of all, let me show you the orientation. We're looking at the brain here from the frontal view. So you're basically looking at this from the front, and what you see in the blue would basically be your ventricles and the central canal. And this is your lateral view. Basically, you're looking at the brain from this view. So what you see as this kind of horn-like structures, kind of like a what do you call it? Oh my God, a goat horn, a ram. Thank you. These little these structures, the semilunar structures, which are actually on the side view look like this. These are referred to as your lateral ventricle. So they're on the side. We refer to them as lateral ventricle. The area that's going right in the middle. So you literally, if you take your cerebrum and divide it into half, it's smack right in the middle. And if you look at it from the side view, it kind of looks like a skull of a bird to me. Do you guys see that? Here's the eye, here's the head, here's the beak, right? This would be your second ventricle on our list, but it's actually called the third ventricle. So the bird head is their third ventricle. So lateral, third ventricle. Then you basically start to go down into your brain tissue. So this structure right here. which is this extension right here called the cerebral aqueduct. So like aqueduct, like the things that carry water, right? The channels that carry water, same thing, same concept here. So cerebral aqueduct. And then what you see right here as this kind of a bulging structure from the frontal view is this structure right here, kind of that little triangle that you see, called the fourth ventricle. So we have lateral ventricle, third ventricle, cerebral aqueduct, and fourth ventricle. And then continuation of the fourth ventricle basically again goes down the vertebral column and form what we call the central canal, which is the little structure you see right here. So be comfortable, guys, when we're looking at this picture. If I give you the picture, would you be able to identify your ventricles on that picture? Would you be able to identify your central canal on it? Okay, now what we're going to do is focus on the brain structure. So far, all I've done with you guys is just show you where they are, what's the fluid around the brain, what are the cavities inside the brain, what are the cavities function, etc. We haven't really talked about the brain itself, right? We haven't really talked about, okay, what does cerebrum does? What does cerebellum does? What is your thalamus, hypothalamus, brain stem, midbrain, pons, etc. do? So for the next part of the PowerPoint, which is the majority of the PowerPoint from this point on, we're actually looking and focusing on individual parts of the brain and associate a specific function to each of them. So let's just start by looking at the largest part of the brain, which is again 80% of your brain, and that's your cerebrum. So we're going to kind of go back to breaking down the cerebrum to little parts and associate specific function to that. So first of all, cerebrum, also the cerebral cortex, which is again the gray matter, the superficial aspect of your brain, is the most rostral, which is the higher region of the brain. And you can see that this is basically your cerebrum. It contains two hemispheres. So again, we already know, if you have ever looked at the brain or ever had a brain for Halloween, we know that there's two hemispheres. There's a line right in the middle. So you get the right hemisphere and the left hemisphere. These hemispheres contain about 80% of what we consider your brain mass. And at the center of your cerebrum, if you guys look at the first picture, again, that I showed you of the brain, smack hidden underneath the cerebrum, you have your diencephalon, which is your thalamus and your hypothalamus. And then following that down, you have your brain stem with the three parts in it. Now, we also mentioned the word cortex before. I typed it in the chat box for you guys. What is the cerebral cortex? Cerebral cortex is going to be that little thin layer, right? The little thin layer at the superficial aspect of the brain, which is made up of, again, gray matter. Keyword here is gray matter, which is the cell bodies. I didn't mention the dendrites, so you don't need to know it. Cell bodies, and then short, non-myelinated or unmyelinated neurons or fibers. So again, if you don't recall this, go back to your chat box or go back to when we're showing the gray matter versus white matter for that. Now, what are these patterns and lines that I see on the surface of the brain? What are the cuts that I see that are more deep, etc.? So let's take a look. So what we have is what we call sulci. Sulci is a plural version. So sulci, if we're looking at the singular version of this, is the same thing as sulcus. So if I use the word sulcus or sulci, it's basically plural version versus a singular version. And these are basically the grooves, the lines that you see on the surface of your cerebrum. Now, if these lines are like super deep, which is literally cutting your brain to pieces, these are called fissures. So sulci are very superficial, they're tiny lines. So look at this, I'll show you this in a picture. So look, this is a sulcus, so this line is not too deep. So that's a sulcus. But if the line is really deep, let's see like how deep it's going into the brain, we refer to that as a fissure. Now, every time I have a sulcus or I have a fissure, you create these little bumps or patterns on the surface of your brain. So the twisted ridges that are forming on the surface of your brain because of the sulci are called gyri. And again, gyri is the plural version. If you're talking about singular pattern, that's called a gyrus. So gyrus versus gyri. The other thing we have is what we call lobes of the brain. So if you guys recall, what were, and I'm asking you this question, so please try to give me an answer. What were the main bones of the cranium that forms basically the curvature of the skull? There are four main bones that form the curvature of your skull. So the frontal bone, right? What else? So frontal, that bone was right here. It ended somewhere right here. What are the bones right here? Parietal bones. On the side are temporal, and on the back of your brain, which are back of your skull, occipital. Correct. So keep that in mind, guys. So if you're looking at the skull, you have frontal, parietal, occipital, and temporal. So based on the location of these brains, or I guess I should say cranial bones, you have the same lobes underneath it. So what does that mean? So if you look at, and I'll come back to this in just a second, guys. If you look at this part of the brain, the front part of the brain, we call that the frontal lobe. This part that is depicted in blue color, light blue color, that's called the parietal lobe. Back of the brain is called the occipital lobe. And the side of your brain is referred to as the temporal lobe. So again, the name of the bone, the name of the lobes of the brain kind of corresponds to each other. The only thing that you don't see by looking at the brain itself is additional folds inside the brain, still part of the cerebrum, but underneath the main layers. And this is this one. So if I pull the frontal lobe and the temporal lobe aside, literally there is a fissure there, I can open it up. I can see additional fold underneath it, and that's referred to as the insula. And that's deep inside your brain, guys. It's still there, it's still part of the cerebrum, but again, it's underneath. So total of 5 lobes, 4 of them associate with the skull bones, and then 1 underneath that, that's insula. So let me make sure I covered everything. Okay. Here's a picture, guys. So looking at your brain, there are also main fissures that basically separate your brain to little pieces. So one of the ones we already kind of discussed, but I didn't give you a name, is the one that gives you your 2 hemispheres of the brain. So the red line that you see right here literally cuts your brain to 2 halves or hemisphere, right and left. And that line is referred to as the longitudinal fissure. Another line that you have, which is very pronounced, and again, you can actually, if we're dissecting a sheep's brain, you could have seen that, is the line that literally takes the cerebrum and separates it from the cerebellum. So you have cerebrum and cerebellum separated, they're not connected, you can open them up. And that line is referred to as the transverse cerebral fissure. So this line that you see, the red line you see right here. So these are the 2 main fissures that we have in the brain. Now we also have 2 main sulci that I need you guys to know. And these sulci are important because they help you see insula, which is the one that you see right here, called the lateral sulcus. And then the next one is this line you see right here that is separating the frontal lobe from the parietal lobe. And this line right here, this sulcus, is called central sulcus. Central sulcus. Now, remember this, we're going to come back to it. We have very specific functions for this, so just learn the name now, familiarize yourself now. The area that's immediately in front of the central sulcus, so this little structure you see in yellow, this part right here, this is the area where it's part of the frontal lobe, but it's also a gyrus, the pattern that you see. Because it's in front of the central sulcus, it comes before the central sulcus. We call it what? Pre-central gyrus. Everybody remember the word pre versus post, right? Pre means before, post means after, right? Pre, like pre-op versus post-op, right? If you have surgery, you have pre-operation, post-operation, same concept. So if you're looking at this, this is before the central sulcus, so I call it precentral gyrus. And this one is after the precentral gyrus, so it's after the central sulcus, so I call it post-central gyrus. So important again, I want you guys to remember, because these are like typical exam questions. Precentral, and it's on the PowerPoint, but I want to make sure you guys get it. Precentral gyrus is part of the frontal lobe, and post-central gyrus is part of the parietal lobe. So precentral gyrus is part of the frontal lobe versus post-central gyrus is part of the parietal lobe. It does make a difference, and you'll see that in a second why that is the case. So far we looked at, in terms of your cerebrum, we looked at the fact that there are shallow grooves called sulci. There are deep grooves called fissures. There are 5 lobes: frontal, parietal, occipital, temporal, and insula. We have looked at the fact that the central sulcus separates the frontal from the parietal lobe, and you have areas in front and the back called precentral and post-central gyrus. We have looked at the main fissures, which is the one that divide your hemispheres to right and left, called the longitudinal fissure, and the one that separates the cerebrum from cerebellum, called transverse cerebral fissure. But again, we haven't really talked about what are the functions of each of these lobes or each parts of your brain. So we're going to dive into that right now. So this is where it gets complicated, guys. So please pay close attention. And if you have any questions, let me know. Now, before I do that, I want to just kind of show you this picture because I think it's a fascinating part of your brain. You can see the color coding here. That depicts how much energy or how actively parts of your brains are. Anything you see in black basically means they are inactive. When you see green color, it means little activity. Yellow means more activity. Orange, increased activity. And where you have these really red colors, that means really high activity. So keep that in mind, and I'll come back to it in just a second. So when we're looking at our cerebral cortex, again, we're just talking about, guys, that little superficial aspect of the brain, just the gray matter portion of the brain. What do we do with your cerebral cortex? Extremely important, guys. Please, please, if you are following me on a PowerPoint, do something for yourself, highlight it, whatever you need to do to remember it. You have 3 main functions for this cerebral cortex. First, you are collecting the sensory data. So Sensory data are basically your sense of touch, smell, taste, hearing, vision. Those are your common senses. So all of the data, okay, let me type this for you guys in Word so you guys can. Oops, sorry, my mouse doesn't work properly. Oh. 1 second. So when we are looking at cerebral cortex functions, please make sure you guys are clear on this. First, collection of sensory data. And this is data that comes from inside and outside. So outside data are basically your vision, your touch, your taste, your sense of a smell. What do I miss? Hearing. These are your outside data, and inside data are things like sense of visceral, which is pain and pressure with your organs, and vestibular, which is your sense of balance. So when I'm talking about collection of data, guys, data is coming from both outside your environment using your 5 common senses, and you also collect data from inside, things like pain and pressure. So if you put a joint in a wrong angle, if your bladder is full, if your stomach is hurting, all of these are visceral senses. And then, where is my head located? Why do I feel dizzy? That's your vestibular sense. I'm saying this is internal. So one main function for the cerebral cortex is data collection, guys. All of the sensory data from inside and outside are associated with your cerebral cortex. Second number, association. Association basically means, simply put, guys, make sense of the data collected from inside from both inside and outside. So example, what I see, what I hear, what I taste, and etc., right? So I know I can hear, for instance, but what am I hearing? Am I hearing music? Am I hearing my phone ringing? Am I hearing somebody talking? What I see, making sense of it. Okay, I can see, but what I'm seeing, am I seeing a person? Am I looking at TV? Am I looking at my phone? Making sense of what I see. So that's the second function. Third main function that we have is associated with voluntary. The keyword here is voluntary, guys. Voluntary motor response, which simply means control of your skeletal muscles only. So I'm doing 3 things with 3 main functions: collection of the data, making sense of that data, and then creating only and only a skeletal muscle response. So I'm going to give you an example. I see a glass of water, like the picture I showed you. Ooh, I'm making a decision. I want that water. What do? I use my skeletal muscle to grab the glass and drink the water. The 3 functions: collection of data, making sense of the data, and eventually do the actual action for a skeletal muscle only. So cerebrum or cerebral cortex have absolutely nothing to do with my heart, has nothing to do with my with my smooth muscle, has nothing to do with my stomach or intestine, has nothing to do with my pupil, has nothing to do with my respiration. All of these are involuntary. I cannot go near them. Only and only a skeletal muscle movement can be controlled by this. So next thing that we have is something that you probably have heard before in terms of your brain, and is the concept of contralateral. Contralateral means opposite, guys. So I'm going to spell this out because sometimes people just write contralateral means opposite. But contralateral in terms of your brain implies that the left side of the body communicates and controlled by the right side of the brain and vice versa. So again, what does that mean? If I'm using my right hand and I'm touching a surface, my data from my right hand is going to the left part of my brain. If my left part of the brain tells me to move, the left is going to cause my right hand to move. Okay? So if somebody, pay attention to this, guys. If somebody has a stroke on the left side of the brain, the right side of the body is paralyzed. If the stroke is on the right side of the brain, the left side gets paralyzed in terms of both inability to collect data probably and basically getting the senses. Can I answer any question for you guys? Anything needs clarification? Please type your questions, guys. We'll finish this slide and we'll stop. So the concept of contralateral in terms of a stroke is if a person has a stroke on the left side of their brain, the right side of the body is affected. So the right side basically becomes paralyzed, or you can call it collect data from it, etc. And vice versa. If a person has problems on the left side of the body, like their face is drooping, they can't move their arms or their legs, that means that the right side of the brain has been damaged by that stroke. Does that make sense, Kristen? Okay. So two other points that I want to make before we take a break is the concept of lateralization. Lateralization basically means that we have each of the cerebrums have their own kind of function. So for instance, a lot of you guys have heard of that. Like you say, the right side of the brain is the emotional, creative side of the brain, and the left side of your brain is the more logical, right, making decision type things. So that's what the word lateralization in a very, very, very broad sense implies. We'll talk about that briefly as we go through the PowerPoint to make this a little bit more easy to understand. But I'll talk about that later. And then lastly, if you look at the cerebral cortex as a whole, you have conscious behavior. That entire cortex is going to be involved one way or another. So what I meant by that is the picture I showed you earlier. So seeing, I'll just kind of explain this to you. We'll see that in a few slides as well. If I'm talking about my sense of vision, guys, sense of vision, the part of the brain that is going to be mostly active, so it's going to do the majority of the work, is going to be your occipital lobe here, okay? Here's the thing though. Just because the occipital lobe is super active, it doesn't mean the rest of the brain is silenced. You're still going to involve the rest of the brain to help you with processing the data. However, majority of the data is going to be processed and handled by the occipital lobe. When you hear something and you're processing that, that's done by your temporal lobe. You can see the temporal lobe lighting up like crazy. But again, the rest of the brain is also show activity. So it's not like the rest of the brain is sleeping and I'm only actively involving the temporal bone. Speaking will show this. This is part of the frontal lobe. We'll see you in a picture in just a minute. And this is the one I like, guys. If you're actively thinking, so if you're using your brain, look at the amount of effort you put in. Everything is light up almost, right? Which means your whole brain is actively involved in the process of thinking, which is a really cool factor. Okay, so let's go ahead and stop here. So it is 9:17, so be back by around 9:32, 9:33, let's say 9:33. So 9:33, be back. We're gonna continue the PowerPoint and we're gonna look at each of the hemispheres on their own, and then we'll go into a more in-depth part of the brain. Oh yes, thank you. I was just thinking of that. So I'll see you guys at 9:33, please. So for your sense of touch or somatosensory, or most of your touches, most of your senses, most of your senses, we have two areas. We have an area that basically receives the data, as I mentioned before, and an area where the association actually takes place, which is basically making sense of that data. So the same concept applies here. So for primary somatosensory cortex, again, the key word here is the primary somatosensory cortex. Put those three words together. The specific location is the post-central gyrus, which is, if you guys go back to your picture, I'm going to kind of go back to the original slide. The post-central gyrus was this part right here. Sorry about that. Finally, I sneezed. So this area, the post-central gyrus, this specific area is associated with collection of data from your sense of touch. Be very careful with the vocabulary, guys. So again, what does it do? It receives generally sensory information from your skin, your joints, your muscles, etc. So when we're talking about your sense of touch, if I say, where does that data from your sense of touch goes? The answer should be post-central gyrus, which is part of the parietal lobe. So that's the key thing. Now, when we're talking about primary somatosensory cortex, another word comes into play, and it's the word homunculus. So somatosensory homunculus. So homunculus is basically if you take your post-central gyrus, which is the image we have on the next slide, if you take your post-central gyrus and you decide, or you kind of assess which part of this post-central gyrus receive data from what part of your body, you notice that parts of your body collect a lot of data, and the other parts of the body does not. So what does that mean exactly? So imagine like you get hit, and I always give this example so you guys can make that connection. Let's say you get hit in the face versus you get hit in your arm or on your back, okay? And if I ask you, well, is it hurt more to get punched in the face or punched in your arm? The answer, if you're a healthy individual, would be your face, right? So why is it that it hurts more to get punched in the face than the arm? The reason is that the data being collected from your arms are less data compared to the data coming from your face, which basically means the number of sensory neurons, the number of neurons that are collecting data from your arm or number-wise are less than the one that collect from your face. So when you go to the brain, when you go to the somatosensory cortex, where those data goes, if you're collecting a huge amount of data, then a bigger part of your brain will be dedicated to processing that data or collecting that data. So look at your face, guys. Face relatively is small compared to the rest of your body, but a big portion of your post-central gyrus is dedicated to processing that data. Now, your arm, your trunk, which is the front and back of your body, your legs, your legs are relatively large parts of your body, but because there is not that much data being collected from it, then you basically don't have a big part dedicated to receiving that data. So when you look at, I'm going to repeat this, when you look at your post-central gyrus, which is receiving somatosensory data, the parts that are collecting a lot of data have a bigger portion dedicated to it on your post-central gyrus. The parts of your body that doesn't have that many sensory receptors in them, there is less dedication in your brain to collect that data. It's like a computer. If you're processing a lot of data, you need more space to process that. If you don't process a lot of data, you don't need to dedicate a significant chunk of your brain to that. So by looking at this picture, guys, which part of your body collect a lot of data? Based on this, what do you think? Look at the size versus how much of the brain part is dedicated to this. Which part of your body collect a lot of somatosensory data? Can you give me an answer? Parts. Not your arm. Your arm is not as much. It is, but it's not as much. Your face, correct. So we already talked about the face. What else? Your mouth, correct. Your mouth is relatively small, but again, you're collecting a lot of data. What else, guys? Your hands, correct. Lots of sensory from your hands, right? Hands are relatively small, but look, big chunk of your brain is dedicated to your hands. What else? Another part of your body. There's two other parts. Your feet, yes. And then what's the last one? Nobody wants to say it. Genitalia. Thank you. Genitals, right. So genitalia is also, kind of think about it, it's going to create your next generation. So if there is something wrong with it, if there is any pressure or pain, you want to know, right? So compared to the size of the organ, right? So in this case, they're showing the male reproductive organ. It is also same true for females' reproductive system. You have a lot of receptors, and therefore a big chunk of your post-central gyrus is dedicated to processing that data. So if I ask you guys, again, listen to the question, which part of the body is going to be more painful if you get hit in, the areas that are going to be really painful, because it has a lot of sensory, based on the pictures, genitalia, feet, hands, or if there is any problem, I guess not only pain. I'm going to say it one more time. Genitalia, feet, hands, face, and your mouth. And then also, if you think about your mouth, your throat and your tongue also includes it. So all of these, basically, these structures, your hand, your feet, and genitalia are included. Is this making sense to you guys so far? Is there any question, clarification on this? I don't see any questions. So if you do have questions, guys, type it and I'll come back to it and I'll answer it. That's fine. Okay, so again, if you look back at the chat box we had earlier, we said there's three main functions. One is collection of the data, right? The second thing I want to do is, okay, so the data I collected, what does that data exactly mean, right? So I collected sensory data from all over my body, right? Somatosensory data from all over my body. Now I need to figure out what that data means, right? So that is called association area. I'm going to associate it to what we have already seen, heard, etc. So in case of somatosensory association cortex, the location of it is right here. So it's part of the parietal lobe. It is in a light blue color. And again, it's called somatosensory association cortex. What does it do? So it integrates, which basically means process, analyze, make sense of the sensory input from the primary somatosensory cortex for understanding of the object. For instance, if you put your hand in your pocket and you feel something is in your pocket, figuring out the size, the texture, the relationship to everything else that is in there helps you, for instance, figure out that, oh, I'm touching my phone, or I'm touching my keys, or I'm touching, I don't know, your wallet. Making sense of the data that you have is the association function. So please, please, guys, this is the most common mistake I see when I ask the students questions. When I ask you, pay attention, when I'm asking you primary blank cortex, whatever that blank is, your sense of vision, auditory, or somatosensory, primary blank cortex, what that means is that it is collecting data from that sense. That's it. It's just collection of data. If I say blank association cortex or blank association area, as soon as you hear the word association, come to mind, it needs to make sense of the data. So if I'm doing this and I'm touching this, right, from my previous experiences, I can figure out that I'm touching my phone because I have recorded that something that textures like this and it has this smooth, cold surface, that is my phone. If I'm, for instance, touching something like this, and even if my eyes are closed, from my previous experiences over my 30 plus years of life, I know that this is what? This is a pen, right? So I'm using my previous knowledge to make sense of what I'm touching. Any question, guys? Okay. So let's apply the same concept to your sense of vision. So sense of vision is associated with the occipital lobe. So we're talking about the back of your brain now. So again, we're talking about primary blank cortex, in this case, visual. So this area, this tiny area that you see in the farthest back portion of your brain in dark blue color is identified as primary visual cortex, which means data is coming from your eyes through the optic nerve in the back of your eye, and it's going to go all the way to the back of your brain, and it's going to basically give you the ability to see. That's it. It's giving you the ability to see. I see a pen. I see I see these objects. Okay? That's it. That's all it's doing. The area immediately in front of this, so this area right immediately in front of that is called visual association area. So what I'm going to do now is I'm going to use my previous knowledge over again, the years of experience that I have, is that when I look at this, I can tell you this is a pen, this is a phone, this is my mouse, right? I'm using my previous knowledge to make sense and interpret what I'm seeing. So ability to recognize a face is association area. Ability to read is association area. So I'm going to give you an example to kind of help you guys understand this a little bit better. So pay attention to this again, exam questions on this one, guys. Damage to the primary visual cortex, all the way in the back of the brain, causes what we call functional blindness. Functional blindness. Why is it called functional blindness? It means, what does that mean? There is nothing wrong with my eye. My eye is collecting the data. I am creating the image, and that data is going to the back of my brain. But it's the back of the brain, because it's damaged, whatever, because of the trauma, a stroke, etc. It cannot collect that data. It is unable to collect the data. So the person is blind. Nothing wrong with the eye, but they cannot collect the data that comes from the eye. In contrast, read this, guys. Individual with damage to the visual association area, which basically means the area that was right in front of it, right? This area right here. They can see. So I can hold this in front of them and they can see something is in front of them. They can see the shape of it, etc., etc. but they might not be able to tell you what this is. They cannot comprehend that this is a pen, or this is a phone, or this is a mouse. So that connection is missing. There's nothing wrong with them seeing the object, like they can hold it, they can do whatever they want to do, but they cannot tell you what they're actually looking at. They cannot comprehend it. So I'm going to give you an example. I don't know if I used this before for you guys, but it is the best way to understand the difference also. So there was a person that says that he has been blind since he was born, okay? And then he goes in and he goes to this temple, whatever, church, mosque, whatever the thing is. And then he says, oh my God, there has been a miracle. And he walks out yelling at everybody, screaming happily that I can see, okay? So somebody holds an item in front of the person and said, oh, you can see, tell me what this is. And says, this is a red pen, okay? And the person that asked the question slaps the guy in the face and says, why are you hitting me? I just told you what I see. And he says, how could you tell me this is a red pen if you have been blind since you've been born? The reason, yes, the reason he knew it was red is why. How do you guys know this is a red color? Because since you were born, or as early as whenever your parents kind of started teaching you words, they would hold a red color in front of you and keep saying it red, 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 red. They will hold this in front of you and they say phone, 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 phone. And your brain made the association that if you see something like this, that is what? A phone. If they hold a color like this in front of you, that color is red. If you are, like I'm around my 2-year-old right now, and a lot of time, she gets the color wrong. She still hasn't fully grasped the colors. So a lot of times, like when I tell her, she goes like, oh, it's pink or it's red, and I have to correct her, right? So she doesn't distinguish the colors very well. But by like in a few months, I hope that when I hold something pink in front of her, because I keep saying her pink, 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 she will remember that it's pink, right? But if somebody's been blind since they've been born, there is no way they would know that this is red color, right? Correct? So basically his brain has never made association points. He can see, very fascinating, but he can never say that this is red. So, you know, like those people that there's the new glasses that the colorblind people can use, and they can see color. These people, a lot of them, you have to retrain their brain to tell them what color they're actually seeing, because they wouldn't be able to see those colors before. So they see a beautiful, clear image in front of them with lots and lots of color, but they cannot tell you what color they're seeing until their brain is trained that this color you see is pink, this color you see is red, etc. Are we clear, guys? Any questions? I'm hearing weird sounds in my house today. Sorry. I'm like, what is this? Okay, so the next thing we're going to talk about, auditory association areas and auditory cortex. So your sense of hearing. So if you look at this, again, there's two areas. There's this tiny area that is in dark blue, part of the temporal lobe. And again, if I call it primary auditory cortex, all it means is that the data from my ears are going to that area, going to that part of my brain. That's it. It's not doing anything. It's not interpreting the information. All it's doing is doing what? Collecting the data. Making sense of the data is associated with this part, the one right behind it, called primary, primary, say one more time, auditory association area, which basically tells you what you're hearing is your phone ringing, you're hearing a piece of music, you're hearing somebody talking, etc. Right? So making sense of that data is your auditory association area. And again, auditory is associated with your temporal lobe. This is your temporal lobe here. Olfactory cortex. the olfactory, which basically means your sense of a smell, does not have that distinction that I've described for you in terms of primary and then the association area. Everything is done with one part of the brain. That being said, it doesn't mean that later on, like 10 years, 20 years down the road, we cannot narrow it down to a specific areas. But as of right now, we have what we call primary olfactory cortex, which is located right here, kind of the midsection of the temporal lobe. And the job is to basically consciously be aware of the odors that are around you. I smell something, that's a smell of trash. I smell something, it's coffee. I smell something, it's cookies. Okay, so that whole area does everything to do with your sense of a smell. So that's all you need to know. Vestibular sense, guys. As I mentioned, vestibular is your sense of balance, right? Basically, where your head is located in this space. If you feel dizzy, it's damage to your vestibular cortex, basically, or it's problem with your vestibular cortex. Where is it located? It's located as part of your insula. Remember, insula is hidden underneath your temporal lobe, so you have to move the temporal lobe to see the insula. That's all you need to know there. We don't make it more complicated than that. If I say, what lobe of the brain? I'm going to type the question, so again, familiarize yourself. What lobe of the brain is responsible for the vestibular sense? Then the answer would, oh, sorry, I messaged Brenda directly. Sorry. Do it to everybody. Then that would be insula. Again, you don't have a specific part of the insula that you need to identify. Neither you have the separation between the primary cortex and your association area. Another thing that your insula does is your sense of gustatory. I don't know if you have heard of that one before. The word gustatory implies your sense of taste. Sense of taste is actually relatively weak, so it doesn't really need a big part of your brain for processing. So that's also done by your insula, which is deep to your temporal lobe. And then lastly, you have your visceral sensory. Visceral, again, is senses that associate with the internal feelings, like the sensation of a full bladder or upset stomach, pain in your intestines, stretching, things like that. These are called visceral senses, and that's also controlled by your insula. So I told my students to remember VVG, visceral, vestibular, gustatory. These are your three senses that are associated with your insula. To review this, guys, I want to just kind of go over this so far, what we have learned. If you're looking at the parietal lobe on the top, you have your somatosensory. Again, ability to sense, touch, and then make sense of what that means. Occipital lobe in the back is responsible for your vision. So basically both collecting the data from your eyes and then making sense of what you see. Temporal lobe is associated with your auditory sense, both cortex in terms primary cortex, what you hear, so ability to hear, and then association area, which is basically what I'm hearing. Next, we have your sense of, oh my God, olfactory. Olfactory is also associated with temporal lobe, but farther down. That's basically what I'm smelling, what that smell is associated with. And then insula is associated with your sense of taste or gustatory, your internal senses, which is visceral, and then your sense of balance, which is vestibular. Any question or clarification on what we have discussed so far? No? Good? Okay. Awesome. Okay, so now let's go to the other lobe that I haven't really discussed, because we talked about parietal, occipital, temporal, and insula. So we talked about 4 out of the 5 lobes. The one we haven't talked about is your frontal lobe. So frontal lobe is associated with the third function, which was the voluntary control of your skeletal muscle movement. So voluntary movement, again, only and only, if you guys go back to what I've typed in the chat box previously, is only your skeletal muscles. Now, it itself has 4 major parts. And I'm just going to show you on the picture because it's a little bit easier to describe, and then we have the information in front of it. So here is your central sulcus line that separates the frontal and parietal lobe. So this line right here is the separation. Sorry, one second. I don't know what happened. So here is, again, I'm just going to kind of show you the line right here. That's separating the frontal lobe from the parietal lobe. The area immediately in front of this, which is the kind of the ruby red color you see, that's your precentral gyrus. Precentral gyrus. And this area is responsible for primary somatic or motor cortex, which is basically means it's going to control your muscles, your skeletal muscles. Now, here is again the important concept: homunculus or homunculi, and then contralateral. Homunculus, we talked about this, guys. Remember I said if you have a lot of sensory in your face, a big part of your brain is going to process that. Now reverse that. If I have a lot of neurons that needs to control my skeletal muscle, then I need a bigger part of my primary motor cortex dedicated to creating that response, creating that motor response. So I'm going to give you an example. So give me 1 second. Let's see if you can process it. When you look at the muscles that are on your back, that's going to help you maintain your posture. These muscles are gigantic, okay? And they don't really need any fine controlled movement. However, when you look at your facial muscles, these muscles require very precise movement, which are creating something called micro expressions. You also have, if you think about your fingertips and movement of your fingers, when you're typing, when you're playing an instrument, if you're looking at a surgeon, they need very precise movement. So if for the face and fingers, I need precise movement, then a bigger part of the brain needs to be processing and controlling those movements. Versus the back of my lumbar region or my back, where it has these gigantic muscles and doesn't really need a lot of control, I don't need to dedicate a big part of my brain to that processing. So the same concept applies here. If you look at your facial muscles, right? If you look at your hands, if you look at your tongue that helps you speak, they need really, really fine control. So I'm going to dedicate a big part of my brain to controlling those muscles. The rest of my body, like my arm, my trunk, my hip, my legs, my feet, they don't have any precise movement. So I don't need to dedicate a big part of my brain to those movements. Is this making sense to you guys? Is that logical? Okay. We also, if you go back to this slide, we also bring up the concept of the contralateral again, right? The whole concept of if you have a stroke, the opposite side of your body is going to be paralyzed. So if there is damage to this part of the brain, this is my motor area, the person basically would be unable to move the opposite side of the body, move the muscles on the opposite side of the body. So paralysis, so if I say the person is paralyzed on the left side, what part of the brain is damaged? What would you say? If the paralysis is on the left side of the body, what part of the brain is damaged? It's a question, guys. The right side, thank you. So opposite side, so contralateral and then a homunculus. So here's another cool part, guys. I think this is one of the most fascinating things. A lot of time we forget about them. So if you're sitting in front of a computer and you're one of those people that have typed probably from the age of 4 or 5 and you use this a keyboard, you don't really think nowadays of how you type, right? You put your hands on the keyboard and you type, right? No thought goes into it. When you're driving, you don't actively think, oh, I need to push the gas, I need to push the brake, I need to push the gas, I need to push the brake, right? You just drive, right? Most of the time that may cause accidents. So hopefully you pay some attention to it. But you basically unconsciously, your muscles are gonna do the job. If you ever played instruments, right? Any type of instrument, again, you don't actively looking at the instrument, right? Which string you're gonna hit or which key you're gonna hit. If you are, you know, those people that are like super fast at their job, like they box things or they cut things, and you're like, how are they doing this? But they don't even look and they just do it. So all of those are associated with a part of the brain, all of those actions that I'm talking about, which you don't consciously think about, are associated with the part of your brain called premotor cortex, which is right here in front of the primary motor cortex. So what this is, it is helping you plan your movement. And what that means is basically a staging area for skilled motor activity. It helps you create controlled, repetitive motions without you actively thinking about the process. So when you type, you're actively using your premotor cortex to send messages to your primary motor cortex so the actual action can take place. So here's the thing, guys. If a person, pay attention to this very carefully, guys. If a person damages their primary motor cortex, which is the ruby red part, the first part I talked about, as seen in a stroke, the muscles are typically paralyzed on those areas. Sounds logical, right? The stroke, opposite part of the body gets paralyzed. Now, the problem is, if you go back to this, the problem is that there's nothing wrong with our muscles, right? Muscles are not damaged when a person has a stroke. We talked about this. The damage is to the connection between your brain and your muscles. That connection is severed or basically halted because the neurons are dead, and therefore you are unable to do the movement. Now, if the damage is to the premotor neurons, so the neurons that are in the area that I'm showing you right here in the premotor cortex, which usually it affects both of these areas. So if a person has a stroke, both of these areas are affected. These individuals forget what their brain has already programmed and has learned really well. So they may forget how to brush their teeth. They may forget how to type, how to drive. And for that, can I retrain my brain to gain that information back? And the answer is what? Can I retrain my brain? Yes, you can retrain your brain to do that. And that's basically when a person goes through therapy, correct? It could be both occupational therapy or it could be physical therapy. A lot of times you have to start with physical therapy and then work your way to occupational therapy. So you need to figure out how to move the muscles and then train it to do something really well. Even as a simple task like tying your shoelace that people probably know since they were like 2-3 years old, right? That needs to be retrained for a lot of people that had a stroke and survived it, because that is part of their premotor cortex functions. Questions about this, guys? Okay. I hope this is making sense to you guys. Hopefully all of you guys are following this. It's not I know it's a lot of information, but it is a logical thing. It's not like there is no connection. Each part of the brain has its own function. So the next thing that we have is this area right here called the Broca's area. So Broca's area is basically area that gives us the ability to speak. So it helps control our vocal cords and help our tongue to basically make sound and or a speech. So it's again active process. So you decide when to talk, like your tongue doesn't do it on its own, right? One thing that I do want you guys to know, and I'm gonna type this in the chat box as well, so you guys have that information. So Broca's area, on the right side, I'm gonna retype it. On the left side is responsible for creation of the actual speech. Broca's area on the right side or right hemisphere is responsible for creation of the emotional overtone. So what does that mean? So when I'm talking, so take my speech right now. When I'm talking, one part of my brain, which is for me would be the left side, is basically putting words together in sentences and passing them out. The right side of the brain at the same time, what it's doing, it's giving an emotional overtone of excitement, sadness, happiness, monotone for some people, right? No emotion put into it. So there's two parts. That's where the specialization or lateralization comes into play. They're both associated with the speech. Both hemispheres are associated with speech, but each take up a task. So one hemisphere is taking over the actual speech. The other hemisphere is taking over in creation of the emotional overtone. The area right on top of that, which is what you see in red, is the frontal eye field. It's self-explanatory. It controls your voluntary eye movement. And I'm very cautious about this, guys. Please do note that I'm saying voluntary eye movement. A lot of the movements in your eyes are not voluntary. For instance, a lot of times when you like tracking an object around you, or when you're reading, you don't actively think about moving your eyes. What I'm talking about controlled voluntary eye movement is like, oh, I want to look this way. Like, okay, so this is what I'm seeing. This is what I'm seeing. So you're actively moving your eyes. That's the voluntary movement. A lot of the eye movement we do are involuntary and are controlled by a different part of the brain. Okay, so this picture you guys see, it's going to be in your exam. They are going to be basically same arrows, but you just, same lines, but you're just going to have them with letters A, B, C, D, whatever. And I can ask questions like this. Identify central sulcus, or which letter represents central sulcus. I can ask which letter responds or corresponds to the part of the brain that controls your skeletal muscle movement. Which part collects data from your eye, okay? So based on what you know so far, you should be able to answer these questions. So I'm just gonna type a couple of questions for you guys in the chat box so you guys again have that idea. So what part of the brain controls voluntary eye movement? And then again, in the image, it would be area, an arrow would be pointing to this region. So the answer would be frontal eye field. Another question that I can ask you, let's say, which of the following sections or following part of the really hard to type and talk at the same time for me. Cerebral cortex is responsible for collection of data from your eyes. And then the answer to that would be primary visual cortex. So I can either ask you the actual name, so the answer name would be primary visual cortex, or I could just have a letter here, it says letter A, and you should pick that letter for the answer. So this picture is your friend, guys. You need to understand what each of these words actually means, and then again, which part of the brain is controlled each of the actions. Question or clarification on anything we discussed so far? No, we're good. Okay. So the next part I want to talk about is what we actually depicted in pink. So we have talked about the areas that are in red and blue, dark and light blue, and basically dark and light red. So we're going to talk about the red sections. Sorry, not the red, the pink sections. Apparently I'm colorblind too. So these areas are called multimodal association areas. Again, there are a good chunk of your cerebrum, and they are broken down to 3 parts. There are anterior association area, posterior association area, and limbic association area. So I'm just going to show you what they do. So the anterior association area, just kind of highlighting the area, is this section right here, kind of this U shape or horseshoe shape that you have. It's called the prefrontal cortex, like front of the front, basically. What it does, it's involved in process of cognition, recollection of information, development of personality, working memory. So as you are basically learning information, most of the data goes into this area. Your intellectual capacity, creation of abstract idea, your ability to judge situations, your reasoning, your planning. A lot of the executive orders are associated with your prefrontal cortex. So again, the area that's highlighted here, that's your association area or anterior association area. Development of this part, and I really want to kind of highlight this idea, guys. Development of this part of your brain depends highly on the social environment that you grow up in and the feedback that you receive from that environment. So basically, it tells you how you behave in a society based on the feedback you receive. And fascinating thing about this part of the brain is that it develops at a very young age. And by the age of typically, I would say around age of 20, the development of this part of the brain is done. So the hardwiring of the brain is done, which basically means if you are in your 20s, the chances are the person you are, you're going to remain that person. Because if you want to change, those changes will not take place easily. Rewiring of your brain will not be an easy process. So kind of looking at this, it says tumors and lesions in the anterior association area. Again, this whole area in front, it can cause mental or personality disorders, include loss of judgment. Again, you don't have that intellect to make that decision. You lose your attentiveness because your working memory is affected. And the inhibitions, again, you forget how you're supposed to act in a social setting. So affected individuals, so if you have tumors or trauma with the prefrontal cortex, they may be oblivious to social restraint. So you may do something that is absolutely idiotic. You may use bad language. You may become careless. You may ignore your personal appearance, take more risks. Again, things that are associated with common sense may not work. So I say a lot of people that participate in the movie Jackass, they have already damaged their prefrontal cortex, and that's why they do those things. Sorry. If you're a fan of that movie, sorry. That is, but that is for sure, right? So I like to. I never watched the thing, so I have no idea. Like I just seen scenes of the movie, and I said there is no way I'm gonna watch that movie. My prefrontal cortex is in place, so I'm not gonna watch it. Okay, so let me just add a quick component to that as well. When you look, and this is the way I look at it. So when I came to US, I was a teenager. So a lot of times, especially if you came with your parents, so I don't know if any of you guys have that background. When you enter a new culture, children have a much easier time making the adjustments to the new culture. And a lot of time that clashes with what the parents are trying to teach their kids in terms of their culture. So basically there would be a gap between the generations versus the younger generation and the older generation. And the main reason, and this is actually regardless of if the cultures are different or you're just born into different cultural zone in terms of time zones. So when your parents, like let's say my parents grew up in Iran, right? So the culture of Iran is very different from the culture of US, right? The people are much more conservative. We follow, the government is very dictatorship, things like that, right? You're taught to be scared of the government, etc., right? Things like, and I'm gonna add this because I think it's kind of funny. The concept of burping, it's really bad in my culture. Like you should not burp. Like it's disgusting. Like it's one of the worst things you could do. So here, so in the US, for instance, when I came, people would burp and they would just say, excuse me, and it's done, right? And my parents would get like super offended, right? So, and it didn't make sense to me because I grew up here and by the time I came here, like I wasn't really socially in terms of like matured, I guess. I was like 14, 15 by that time. So it wasn't a big deal for me. So for me, it's like if somebody burps right now, I'm like, oh, okay, fine. I don't like it necessarily, but it's not like, oh my God, I'm offended big time. So the reason for that is because my brain, when I came here, wasn't fully developed. More specifically, my anterior association area wasn't fully developed. So I accepted part of my parents' culture, which was already there, but my brain continued to wire itself here in the new culture. And based on the feedback I got from the social environment, I accepted burping as, it's okay. Just say excuse me and move on. But again, you can see the difference between just that simple aspect of the culture. Their brain is already hardwired that burping is a really bad action. It's so super offensive versus for me, it's okay. It's not the best thing, but it's okay, right? So take that and take it into a much larger things. So a lot of times I talk to my students, they're like, my parents don't understand me, this and this and this. And I'm like, you guys also need to understand, especially if your parents come from a different culture, their brain is already hardwired in a very specific sense versus you guys, you got exposure to both culture, which means your brain wiring is significantly different from your parents or your previous generation, and it's much easier for you to adapt to those cultures. And you can hear that a lot of times people say, oh, it's much easier for children and youth to adapt to a culture versus an adult. Main reason is being the development of the anterior association area, which I find fascinating. So it's not under their control. Don't blame your parents. Yes, you may slightly change, but overall, you will not be changing a person that is in their mid-20s. You cannot rewire the whole brain. So don't expect that, basically. Be patient. Okay, so hopefully that makes sense. Unless you guys have questions, again, please type them in. Okay, the other two association area we have is limbic association area, which is not shown here. Limbic association area is, and I'm going to just read this and then explain it. It says, provide emotional impact that makes sense in terms of what is important to us and help us establish memory. So in terms of what we form as good memories or bad memories, this is the part of your brain that does that. For instance, if you were kids and let's say your grandma used to make cookies and or bake for you or cook for you, and you have this really positive memory associated with a specific smell, then when you grow up, let's say 10, 20 years down the road, and you smell that cookie or you smell the bread or food or whatever that thing was, it triggers a positive emotion in you because you associate it with a positive memory. On the other hand, you can have the same concept in terms of negative memories. So for instance, if let's say you were in a car accident when you were super young and right before the car hit you, there was a smell of burnt tire. Now, every time you may smell that burnt tire in a different environment, you may feel anxious, worried, sad, upset, whatever that emotion is, because it triggers that negative emotion in you because it associates with a negative memory. So hopefully that concept, limbic association area, makes sense in terms of memory. Yeah. That's kind of sad, but yeah, ages, sorry, I'm just reading. I don't know if that was directly to me. But yeah, young kids are significantly different in terms of growing up. So a lot of times they miss a lot of the good parts of the culture that the parents grew up in. Limbic association area is actually not shown in the picture. And I think I might have, if I go all the way to, sorry. I want to see if I actually have that. Oh, I don't have a slide for it. It's central in the brain. It's kind of, it's at the center of the brain. You won't be able to see it on the images that we have. It's not as clear cut as what we have for the anterior and posterior association area. So I'm not going to ask you guys where it's located. I thought I had a slide for it, but apparently I don't. So sorry, we can ignore that. Okay. Posterior association area is basically what you see here as this whole area depicted in pink. And this area is important in terms of you recognizing faces, patterns, where you are located in terms of your environment. And they also have a specific area, which is what I highlighted in, or it's, I guess, dashed line here, called the Wernicke's area. So what you need to know for the Wernicke's area, and I'm going to type that also in your chat box. So Wernicke's, which is the name of the person who actually discovered this. Wernicke's area is associated with understanding a spoken language. So same thing, you have the right and left lateralization. So the left hemisphere within the Wernicke's area is associated with understanding the spoken words. And the right hemisphere is associated with ability with emotional overtone. So again, you guys figuring out as I'm talking that I'm excited, I'm happy, I'm sad, I didn't put any emotion in my thing, that's the right side. For you to understand what I'm actually saying, that's the left side. within your Wernicke's area. So what if there is a problem with the posterior association area? These individuals usually have problem with figuring out where they are located in the space around them. I don't know if you guys have ever heard of these people, like if you have a lesion or a trauma in the posterior association area, you may refuse to wear like clothing for part of your body. Basically said, this is not part of my body. Like they can see it's attached, but their brain says, no, this is not part of you. So they refuse to basically put clothes on because they believe that that's not part of them, which is really creepy because like, can you imagine like the argument that happens inside? Like your eyes tells you that your arm is attached to your body, right? But your brain says, no, this is not you. So that just kind of, that always freaks me out. I mean, however, it is relatively easy to diagnose because of the way the person will start to act. And again, that's very simple. Yeah, to me, it's fascinating. Like it blows my mind every time you think about the brain and how small it is compared to the rest of your body, but how the smallest problem with it can cause significant changes in that person in terms of, you know, what actions they can take all the way to their behavior. Okay. So we already talked about lateralization, which is that kind of, oh, sorry, I don't know why I draw the line. That, you know, right hemisphere does this, left hemisphere does that. A lot of you guys may have heard of the concept of cerebral dominance. So this is basically which hemisphere of your brain is related to your language. So most of the people, and by most I mean 90% of the human, have left side dominance. So basically this side of the brain, the left side, is controlling your ability to create a speech. That means that you are right-handed. So your right hand side is going to be your dominant because of the whole concept of contralateral, right? If you are left-handed, the dominant side of the brain that creates speech is the right side. So I'm going to say it one more time. If you are speaking, right, and you are left-handed, your ability to create speech is done by the right side of the body, right side of the brain, sorry. For majority of us, if you are right-handed, so you write with your right hand, the part of your brain that is actually helping you make your speech is the left hemisphere. So your left hemisphere is the dominant one. So for 90% of the people, the left side controls your speech, the right side controls the emotional overtone. Wernicke's area, the left side again controls your speech, and the right side is understanding the emotional overtone. Yeah, sorry, that's kind of sad. Sorry, I keep reading the chat box. At least it was diagnosed though, so that's a good thing. Okay, so the next part that we're going to talk about is the communication that happens inside your brain. And this communication happens three ways. And I want you guys to not ignore the names right now, but I want to just kind of see the three types of connections. So you have two hemispheres, right? The first connection that you want to have is each of the hemispheres want to communicate within themselves. So frontal lobe wants to communicate with parietal, parietal wants to communicate with temporal, temporal wants to communicate with occipital, etc. So you have communication within each of your lobes. That's one type of communication. Second type of communication is the right side of your brain wants to be able to communicate with the left side of the brain. So you want to have cross connection. Third type of connection is you want your brain to work with the spinal cord and the rest of your body. So the connection is between two organs. So one more time, connection within the hemisphere, connections between the two hemispheres, and connection between the brain and the spinal cord. So what are they called? If they are within one hemisphere, they're called association fibers, which are depicted in red. So again, they're all in one hemisphere. If they are crossing between the two hemispheres, they're called commissural fibers, and they're also called corpus callosum. And these are the green ones that you see. Again, a stretching between the two hemispheres. And this is really important because you don't want your left leg to do something and then your right leg to do something else, right? You want them to work together to make you walk, for instance. Third type of connection, which are called projection fibers, is basically take the two hemispheres as a whole and bring them down and allow the communication with your spinal cord, which is the extensions you see going down the spinal cord. So three types: within, across, between the brain and the spinal cord. Questions about that? How are we doing on time? Okay, good. Okay. So we gone through majority of the function of the brain, since cerebrum was 80% of your brain. So we're going to very briefly, and I really mean very briefly, look at the functions of your diencephalon and your brainstem. So they're not as detailed as what we saw for cerebrum, but they still give you some good information. So diencephalon is made up of two major parts and a kind of a smaller third part that I didn't mention before. That's thalamus, hypothalamus. So thalamus was this big part, this big part, and then this is your hypothalamus. Oh, it looks like a bird right now. And then epithalamus is not really depicted in this picture, but I'll talk about it. Oh, it is actually, sorry, it's right here, right here. That's your epithalamus. So it has really high concentration of gray matter, which means there's a lot of neurons, a lot of cell bodies, a lot of activities. So what is each of these parts going to do? So thalamus, which is again that big oval structure you see right here, it's 80% of your diencephalon, and it's identified as the gateway to cerebral cortex. That's usually how I describe the function. So when you bring data into your brain for all your senses, with the exception of your sense of a smell or olfactory, this data first needs to go to your thalamus. Your thalamus sense what that data coming from, adjust it, like basically say, oh, the signal's too strong, I'm gonna weaken it, or the signal's too weak, I need to strengthen it. Kind of makes this tiny adjustment to it, and then send it in its merry way to its final destination. Kind of think about it as your post office. You send a message from your eye. This message from your eye goes to your thalamus. Your thalamus says, okay, this is that from optic nerve. I need to send it to the occipital lobe. Data goes to the occipital lobe. Data comes from your ear. It goes through the thalamus. It says, this is auditory data. I'm going to send it to the temporal lobe. So it basically figures out where the data needs to go, make the adjustment, and then send it in its merry way. That's all you need to know for the thalamus. Hypothalamus is a little bit more complicated. Even though it's relatively small, so compared to the thalamus, it's a relatively small structure. It does a lot of function, guys. What are these? First of all, and this is missing actually, so please add it. It is the relay station for for olfactory. Did I have that? No. Olfactory sense. So the one that was missing from thalamus is done by your hypothalamus. The other thing that it secretes hormones. So basically it controls the pituitary glands, the one that hangs right underneath it, remember? The things, these little pink and purple circles that hangs from the hypothalamus. You control what hormones gets released from them. It controls your autonomic effect. If you guys remember the word autonomic, it basically implies your fight or flight or your rest and digest response. So it controls those. It has a role in it. It's not fully controlling it, maybe I should say. Thermoregulation basically controls your body temperature. So if you have a fever or your body temperature is below normal, there is a that's the job of your hypothalamus. Food and water intake basically can tell you when you're thirsty or when you're hungry. It's kind of cool. It tells you when to sleep. So we call that your sleep-wake cycle. So a sleep and wake cycle is controlled by hypothalamus. But I do need to add that hypothalamus, and I'm just going to introduce it here. So hypothalamus controls the release of hormone melatonin from your epithalamus and therefore control the sleep/wake cycle. It helps you create emotional response, and it also plays a role in memory. So again, if there is a problem with your hypothalamus, a lot of things could go wrong. Next. We are talking about your brainstem. So brainstem was the three-part structure. The top one was the midbrain, followed by the pons and medulla oblongata. So here is basically sorry, I'm gonna go back to this picture. So right here is again your thalamus. So what you see in green is midbrain. The bulging part you see right here is your pons. And then the last part is the medulla oblongata. So midbrain is important for us in terms of what we call controlling your visual and auditory reflexes. So in the terms of your midbrain, if I look at it from the back, so if basically I'm putting a camera here and look at the back of the midbrain, you have two little structures. So these two that are paired, and then this structure right below it, this one right here. So the top one is called the superior colliculi, and the bottom one is called inferior colliculi, when we're talking about both of them, or colliculus if we're talking about one of them. What it does is the superior colliculi portion of the midbrain is control what we call visual reflexes. So what it means is basically your eye tracking an object is an involuntary thing. Again, remember I said eye movement can be voluntary or involuntary. So the involuntary movement is done by your superior colliculi. So I tell my students kind of think about it in a sense that if you have something in front of you, you don't actively think about, I need to follow this, I need to follow this. Or if you're reading a book, you don't continuously think, oh, I need to follow the line, I need to follow the line, right? You just do it. So that's basically your eye tracking capacity. Inferior collicular is associated with you basically responding to sounds. So when you hear a loud noise, being startled by that sound, and then usually what do you do? If you hear a loud noise, you immediately turn your head toward the sound or the direction of the sound. So that's identified as auditory reflexes. And again, it's done by your inferior colliculi. The structure as a whole in terms of visual and auditory reflexes is referred to as the tactum. Another characteristic of the midbrain is what we call the substantia nigra. So substantia nigra is really important to us for, first of all, let me show you to you guys. It's these structures right here. So these semilunar structures right here. These are basically contain a significant gray matter within the white matter, and really, really important in terms of voluntary movement. So they work very closely with your primary motor cortex. Hopefully all of you guys at some point have heard of Parkinson's disease. Do you know what's the most common symptoms of somebody that has Parkinson's, guys? Anybody? No, nobody? Speech is not necessarily affected as much other than involuntary movement or jerky movement of the, shaking or jerking movement of the limbs. So they basically have uncontrollable shakes. So these individuals, they say that when you look at their substantia nigra, the neurons that are in that part of their body is significantly degraded. So it's not the primary motor cortex that's affected, it's the substantia nigra portion of the midbrain that is affected. Just kind of sad. Another structure we have is these kind of egg-like structure, oval shapes called red nucleus. These are high in iron and blood supply, and they do play a role in flexion movements of your limbs. Again, when you think about the main part of the brain that does that is your primary motor cortex, they have secondary roles in these jobs. Now, when we look at your brain, we have 12 cranial nerves, which basically means nerve that comes from the or associated with brain. The cranial nerves number 3 and 4, so Roman numerals 3 and 4, called oculomotor and trochlear nerves, are associated with your midbrain. I don't need you guys to know what they do. If you are interested, these are both associated with movement of your eyes. So they do play a role in the, again, the voluntary movement of the eye muscles. But what I really do need you guys to know is if I said which nerves are associated with your midbrain, you should automatically say cranial nerve number 3 and 4. That's all I need you guys to know with the proper name. But we don't go into the function. Pons, which is the second portion, what you see as, again, this kind of bulging structure right here. This is brain from underneath. That sits right below your midbrain. It has cranial nerves 5, 6, 7, and 8 associated with it. So again, know the numbers, nothing beyond that. What does the pons do? And I'm gonna add this because this is really clear. When I ask you an exam question, you have to be super clear about this answer. What is the function of the pons? The main function is acting as secondary respiratory center. Secondary, the keyword here is the secondary respiratory center. So that basically means that it does play a role in respiration, in helping you control the rate of the breathing and the depth of the breathing, but it's secondary function, not the primary. It controls bladder movements. So in terms of when you need to pee, when does the muscles relax, the sensation of a full bladder, etc. It helps you with your facial expression. So again, controlling voluntary movement in a minor sense, facial expression, equilibrium, and eye movement. And then the last part that we have, which is the last part of the brain stem, is medulla oblongata. So I'm going to kind of skip this slide and come back. Medulla oblongata is connected to cranial nerves number 9, 10, 11, and 12. So number 9 through 12 is connected to medulla oblongata. So again, if I ask you what is the function of medulla oblongata, your answer should be, again, keywords here, guys: primary respiratory center, primary cardiovascular center, which basically means it controls heart rate, and blood pressure. And the third function that I really want you guys to know is control of involuntary reflexes, such as, again, hiccuping, coughing, swallowing, and sneezing. Oh, and then basically throwing up, so vomiting. So sometimes my students ask me, well, I can choose when to cough, or I can choose when to swallow my food. True, but if you guys, if you look forward, and I haven't talked about it, in terms of digestive system, the bottom part of your esophagus, right before it connects to your stomach, you really have no control over when that food goes down. So that's what we're talking about here. So not swallowing that, like I do this and I swallow my food. I'm not talking about that part. We're talking about farther down, where the food actually goes farther down your esophagus. Same thing with coughing. So, you know, sometimes, yes, a lot of times I control my cough, but let's say you have you have something stuck in your throat, right? Or you have a cold and you have phlegm built up in your air passageway. You can't control those coughs. Those coughs are going to happen. So those are the reflexes that I'm talking about. You definitely can't control your sneeze or vomiting. You can't really swallow your vomit, sorry, but you can't really control it. It's gonna happen. I think, let me go back. Okay, pyramids you can ignore. So this part of the slide, basically you can ignore. I don't need you guys to know anything about this. So you can just ignore. This is one of the coolest things that you have in your brain, and I just want to kind of very briefly go over it because I don't expect you guys to, this is not a physiology class, so we don't really go into too much detail about this, but I think it's really fascinating. It's an area of your brain stem and the rest of the brain called reticular formation. Reticular formation is what gives you your consciousness or your alertness. So basically, if you're sitting here, you're listening to me, you're processing the data around you, you're not daydreaming or in a coma, you're actively using your reticular formation to stay aware of your environment. It is also the part of your brain that gets rid of the repetitive motions that basically going to be inconsequential to your survival. What does that mean? Your ability to focus on a conversation in a very loud room. So if there's a lot of conversation happening around you, but you're having a conversation with the person in front of you, you're going to focus on the person in front of you and kind of ignore all the other sounds. Having your clothes on and not noticing that your clothes are on, right? So imagine you continuously collect data from your senses. Oh, I have clothes on. Oh, I have clothes on. Oh, I have clothes on, right? You don't actively think about that. If the clothes move on your skin, then you notice that movement, but otherwise your brain basically ignores it. So when I talk about reticular formation, it is in two senses. First, getting rid of things that are inconsequential in terms of your health and your survival, and then being aware of your environment. So if somebody basically is unconscious or is in a coma, the reticular formation is knocked out. What does the reticular formation looks like? These are the red dots that you see. They go up through the brainstem, which is the purple structures you see, and then they basically expand all the way through your cerebrum. So there's no specific part of the cerebrum. It literally branches off and spreads through all of the cerebrum, associated with all of your senses, all of your motions. So if you're unconscious, you won't be able to move your muscle. If you're unconscious, you're not really collecting data from your eyes, your ears, your hearing, right? And that's why people in a coma basically have no reticular formation functioning. All right. Next thing you're going to talk about is your cerebellum. Cerebellum is really simple. And again, you don't really need to kind of review this part. So I'm going to just draw an X on it. It does made up of 3 lobes. So cerebellum, like cerebrum, has its own lobes. The lobes are labeled. So you have the light orange color up here called the anterior lobe. The bigger lobe on the bottom called the posterior lobe. And then you have this little lobe right here. Sorry, my thing is just gonna move. Called the follicular nodular lobe. The pattern that you see right here, it's called folia. So in terms of cerebrum, those patterns were called gyrus. In terms of your cerebellum, those patterns are called folia. The way you connect the 2 hemispheres for cerebellum, that connection is called vermis. So here is your 2 hemispheres. So this area right in the middle where the connection happens called the vermis. And the fissures, there is the fissure that basically the deep groove that separates the cerebellum to the right hemispheres and the left hemisphere. So that's all you need to know. Now, what is the function of cerebellum? I'm going to type this in the chat box as well. What is the function of cerebellum? The main function is creation of controlled and smooth motions of the skeletal muscle with the help of the primary motor cortex. So when you walk, guys, basically what I mean by this, when you walk, you don't walk like a robot, right? It's not like your motions are very kind of rough, right? Your motion is very smooth. Like you take one step and then another step and another step. You don't actively bend your knees, lift your leg, and then put it down, right? That beautiful motion, that is smooth motion, that's a smooth transition between your movement, is your cerebellum working with your primary motor cortex to create those beautiful movements. And I always just throw it out there that, you know, in children, when they're born, their ability to create any type of a skeletal movement that are clear and concise and smooth is basically non-existent. As they grow, they learn how to crawl, right? And when they crawl at the beginning, they're kind of like, you know, those tiny robots that are running out of battery. And then when they start to walk, they fall a lot, right? When they walk, they kind of don't bend their knees, right? So all of these changes that you see as the child starts to go through their developmental stage happens because their cerebellum is continuously developing. So as the cerebellum develops, the movements of the skeletal muscle becomes more smooth. So that's the main function of your cerebellum. You can ignore all of this. All you need to know again is what I've already discussed with you guys, nothing beyond that. Limbic system, we already kind of described this. Limbic system is your, oh, this is the slide I was looking for actually. Somebody asked me where is it located. So it's the orange area that you have right here. That's basically your limbic system, which is the limbic association area as well. There are 3 main parts that you need to know for this. First one is the cingulate gyrus. Cingulate gyrus is important, is expressing emotion through gestures. So when I do this, I'm actively using my cingulate gyrus. The other part that you have is the hippocampus, which is the area. Please mute yourself, whoever it is. Hippocampus is the area right here. This is helping you create long-term memories. It helps you create your behavior based on your environment. And the whole idea of creating or connecting emotions to how you would act when that emotion basically pops up. So if you're happy, you're gonna smile, your body's gonna relax, versus when you're stressed out, your muscles are gonna tense up. You may be angry, you might be lashing out. So all of those are associated with your hippocampus. Amygdala. Amygdala is the part that you see right here. It's that little circle right here. It's the part that processes fears and memory based on the emotional impact. So for instance, if you have heard of a sound, and again, that sound was something connected to a horrific memory, if you hear that sound, the chances are you're going to avoid that environment. You're going to try to run away from that environment. That's basically what that thing is. I used to say that actually, the Italian use their cingulate gyrus a lot, but I was like, maybe I'm offending people, so I stopped saying it. But yes, that is true. Sorry, so no offense to Italian, but I sometimes I think I'm like literally 90% Italian because I can't do without my hands. Like I have to use my hands. But anyway, so for the limbic system, know these three main parts. There's a lot more parts into this limbic system. It's a really complex system, but these are the most important one that you need to know. All right, we're almost there, so bear with me for a couple more slides. Spinal cord, which is the next part that we're going to talk about, which is part of the central nervous system. It is associated with, or in terms of function, it acts as a highway between your brain and the rest of your body. So basically, if the data needs to come into your brain, it needs to pass through the spinal cord. And then if the data needs to go out, which means in terms of motor, it goes through the spinal cord and comes out of that to go to the rest of the body. It also, as I mentioned at the beginning of the lecture, it is acting as the reflex center. So if you touch something hot or you walk on a surface that is cold or sharp, that reflex of immediately pulling away your limb, that has to do with your spinal cord, not your brain. You have total of 31 nerves. So you have 12 nerves for your brain and 31 nerves for your spinal cord. They're both front and back, so dorsal and ventral, that's basically what it means. And their location basically decide the terminology we use to name them. And I believe I have a slide on that. So if they're associated with the neck region, we call those cervical. If they're associated with your thorax or your basically your chest, they're called thoracic. They're 12 nerves. Your lower back, they're called lumbar. The sacrum, the bone sacrum, if they're associated with that, they're called sacral. And then if they're associated with your tailbone, we call it the coccygeal nerve. A lot of people, or some of the textbook, maybe I should say, they put these together and identify it as the sacrococcygeal nerves and just put 6 for them. Depending on which book you're talking about. But again, that adds up to 31 nerves coming off of your spinal cord. Know the numbers, guys, know the locations. That's it. Woo! Only went 2 minutes over the time. Is there any question or clarification that you guys have about the concept we described? I know it's a lot of information, but it's also a very cool lecture to me. One of the coolest lectures we do.